in today's InnoTalk, we're honored to have Dr. Daniel Weiss with us. Dr. Weiss is the Chief Scientific Officer of International Society for Cell and Gene Therapy, as well as a professor at the University of Vermont School of Medicine. In this webinar, Dr. Daniel Weiss will share his insights and discuss upcoming advances and opportunities in lung regenerative medicine, the current state of cell-based therapies in delivering therapeutics to the lung, as well as the use of bioengineering method to improve lung disease and more. Now, let's welcome Dr. Daniel Weiss.
All right, Ella, thank you very much for the opportunity to work with you and to present today. And I'm going to cover a lot of territory because there is a lot of exciting work going on in the regenerative medicine and regenerative engineering field in respiratory disease and respiratory health. So just as a quick additional background, I'm a critical care physician at the University of Vermont. So I spend part of my time taking care of patients in the intensive care unit, including COVID-19 patients. But I also run a research program in lung regenerative medicine. And so what I'm going to do is show you, again, how this world has come together and where things can be going with this. So what's the goal here? You see a schematic of an emphysematous lung with holes in it from cigarette smoking and other disease and the like. And our goal is to try and figure out how to fix this lung, both structurally and functionally, so that we can get back to something that is useful for a patient, so a patient can breathe again. And there's a lot of work going on in a variety of spectrum approaches, if you will. But what I want to cover today, first of all, are the advances in ex vivo tissue and organ bioengineering. Can we grow functional lung tissue out of the body that would be eventually useful in transplantation or replacement of a damaged and diseased lung? And then we're going to talk about functional repair and immunomodulation. And this will get into cell-based therapies and will cover, in particular, COVID-19. So with that in mind, why do we do this? And the problem is that most lung diseases remain incurable. There is no cure for emphysema. There is no cure for asthma. There is no cure for cystic fibrosis. There is no cure for idiopathic pulmonary fibrosis. We have medications and we have treatment strategies that can help to improve quality of life and hopefully slow the progression of each of those respective diseases, but there is no cure. In parallel, we deal with critical illnesses. So things like the acute respiratory distress syndrome, which we'll talk about a bit later on, but is the main killer, if you will, in COVID. And so there are no cures. We have, again, supportive care, but we're desperately looking for new therapeutic approaches. And it's something that I need to point out right above, and I hope you can see my cursor here, that there is no one size fits all. So we should be on a slide in which the title is Lung Disease, Critical Illness, Targets, or Novel Approaches. Is that correct? All right, very good. All right, so just to quickly recap what we just said, the unfortunate fact is that most lung diseases and critical illnesses are incurable, and we need to find new approaches, but we need to match the approach to the disease pathophysiology. And so when you think about a breakdown in terms of the diseases we're going to be dealing with, there are the acute inflammatory diseases, and those are marked by high intensity transient inflammation with inflammatory and immune dysregulation. And these are things like the acute respiratory distress syndrome, when the lungs essentially go bad on you. And this is part of COVID, but also diseases like sepsis and septic shock. Then we have a broad category of chronic inflammatory diseases, including things like asthma, cystic fibrosis. In the pediatric world, diseases like bronchopulmonary dysplasia. And then we've got the chronic fibrotic or destructive diseases like COPD, chronic obstructive pulmonary disease, idiopathic pulmonary fibrosis. So let's see, hopefully this will work. Do you see the next screen right now with lots of pretty color schematics? And
yes.
All right, very good. Okay, so these are the approaches that I want to go over with you, the approaches we're taking in my laboratory. And so again, the core in the center here is how do you fix a broken lung? And so in the upper left-hand corner, because can you see the cursor or not? You see the cursor, all right, very good.
Yes.
Oh, good. So we'll talk somewhat about stem cells and organoid cultures and the variety of cells that can be utilized for these studies. In the upper right-hand corner, we'll talk about cell-based therapies, in particular using mesenchymal stromal cells for ARDS and COVID ARDS. We'll talk about that a bit further on. But we'll get going with some of the lung bioengineering. And can you grow new lungs outside of the body? Can you make lung assist devices that will supplement lung function? We'll get into some of the more engineering-based applications with hydrogels and three-dimensional bioprinting, and then novel pleural sealants, a spinoff that has immediate practical use. So there's a lot going on. So if you want to engineer a lung, if you want to grow a lung outside of the body, what's the primary function of the lung? It's gas exchange. You all remember, breathe in, oxygen's got to go from the lungs to the blood. Carbon dioxide's got to go from the blood back to the lungs to get exhaled. And this is the primary goal of any device or artificial lung. And it's a challenge. The lung is a very complicated, structural, functional related organ, as you can see in this schematic in the upper right-hand side. It's also cellularly a very complicated organ. And in the lower right-hand schematic is just an approximation of what are felt to be roughly 40 different types of cells that make up the structure of an adult lung. And so a very complicated system to work with. Lung also acts as a filter to prevent particles from getting into the body. And then in terms of other design requirements, you need certainly long life cycle, immune compatibility. Lung also has some inflammatory or anti-inflammatory functions that need to be considered. So if you want to build a new lung, you got to start somewhere. And so you need a scaffold, right? You need some sort of erector set. And ideally, you would add organ-specific cells. So you'd add lung cells, or you'd add stem cells that would then be able to differentiate into functional lung cells. Ideally, you'd want these from the patient who's going to get that lung transplant or that lung you've grown outside of the body to minimize any immune mismatch or misrecognition. And so the idea, the Holy Grail, is to tailor make new organs for transplantation. And so if you think about it, this is one of the ultimates in personalized medicine. So how do you do that? Well, there are all types of technologies that have emerged over the past number of years. One very powerful technology is what's called lung on a chip. And you've probably heard about organ on a chip technology. So it's the same for lung. And you can make a microsystem in which you have microfluidic channels. And on one side, you've got airway epithelial cells, the airway lining cells of the lung. On the other side, you've got the vascular endothelial cells. And so this would mimic that gas exchange system that is part of normal physiology. And then you'd have some sort of matrix in between the two separating them. And these chambers can be very sophisticated because you can alter the flow of fluid through the vascular side. You can blow air in and out of the airway side. You can stretch these membranes, if you will, to mimic the dynamic motion of breathing. And they are very powerful tools, and they're particularly powerful tools for drug screening. But no one is going to grow a new lung out of a lung on a chip. So tool, but not quite there. What about 3D bioprinting? So there are many advances in 3D bioprinting for a variety of different organs, and these clever schematics of growing new ears and new noses for cosmetic and functional replacement, if you will. Very powerful, but right now the resolution is not quite there for the level of the alveoli, if you will. It'll be there soon enough. And combination of 3D bioprinting with techniques such as stereolithography and others are going to get us there sooner than later. But it'll be an amazing day when someone actually 3D bioprints functional lung or functional lobe of lung. So keep your eyes and ears posted for that. But this is definitely a growth area, if you will. Now, part of the problem with 3D bioprinting is what do you use for the material to 3D print that lung? And that's an open question. How do you get the cells into the right place in the lung Do you want cells that belong in the airways to somehow end up in the alveoli and vice versa? So there's what we call a zip code approach in trying to design how to best make this 3D bioprinted lung. So these are all good techniques, but it turns out that the ultimate, and just to go off on a little bit of a tangent, the respiratory system includes not just the lungs themselves, but of course the airways, and the major airways, of course, is the trachea. Now, tracheal diseases compared to lung diseases aren't as common, but there are certainly congenital, devastating congenital conditions, trauma, including battlefield trauma, cancer and the like. And so there have been advances in 3D bioprinting, and in that lower left-hand schematic is an actual 3D bioprinted trachea using computer guidance, CT, CAT scan guided computer guidance that some very clever investigators at the University of Michigan produced, and actually inserted this into a neonate with a congenital tracheal defect and saved that neonate's life. So this is incredible. But part of the challenge, and that's not yet been bridged, is, well, that neonate's going to grow up. And so we need materials that are going to grow as the infant grows. And so you can appreciate the beauty of the advance, but the challenges that still remain. Other techniques, I just show you one in particular, using, again, engineering approaches, electrospinning, to create new biomaterials to use for tracheal replacements, if you will. But what about the lung? Right, lungs are very complicated, and it's tough to 3D bioprint or electrospin. So it turns out, when you go back and think about scaffolds, that the lung itself is its own best scaffold. And many of you may have heard about decellularization of different organs and tissues. And the idea here is that one can take an organ and perfuse it, infuse it with a variety of materials, such as detergents, hypertonic solutions, hypotonic solutions, DNases to break down DNA, with the goal of removing all of the resident cells and cellular debris. And so what you're left with is the underlying three-dimensional scaffold of the lung. And so what you see in the upper left-hand corner here is a picture of a mouse lung. And you can see catheter, needle, angiocath in the trachea. You can see the heart is still attached to it. But then you see this pearly white tissue that is the mouse lung that's had all of its cells removed. And it maintains grossly the appropriate structure. Microscopically, these are some representative electron micrographs showing trachea, the proximal airways, distal airways, and alveoli, so that you retain the ultrastructure as well as the gross structure. And we can do all types of clever things in terms of perfusing the lungs. So here's dye, blue dye going in the pulmonary artery, extending throughout the lung, and then coming right out the pulmonary vein. So we've maintained it in intact vascular channels. We can ventilate these lungs, and then we can use all types of different bioreactor chambers. And this picture is deliberately meant to show you the complexity of, here's the lung in a plexiglass chamber hooked up to all types of pumps and ventilators and the like too. So we can keep these lungs going for quite some time. All right. And then the challenge is, how do we make the best of it? So here is, for example, a native lung or a lobe of lung. And then we go around the circle here. We have to decellularize, and there is no one right way. And so there are a variety of physical, enzymatic, chemical approaches to do that. And then we want to see what's left over. So the characterization of the scaffold, the 3D structure, did you get all that DNA out? What's the composition of the underlying ECM, extracellular matrix, that's left over? What are their mechanical properties? And then what kind of cells, when you go now up the stream to recellularize? So do you use differentiated lung cells? Do you use progenitor cells such as ESCs, embryonic stem cells, or IPS, induced pluripotent stem cells? And so there's a wide menu to choose from. And then again, you have to be able to get the cells into the right place. And then even more complicated, do you put them all at the same time? Do you do them sequentially? What growth media do you use? What oxygen do you use, the PO2, what temperature? And then this, because this is lung, cyclic stretch. Do you take these lungs and you ventilate them to different degrees of frequency and amplitude to try and mimic the best environment that's going to sort or support regrowth and the like. And then at the end of the day, you want to implant and show gas exchange evaluation. So there's been a lot of work in this going on for the past 10 years, and we've reached some very good levels where we're almost there, but not quite there. So this is, again, very exciting technology, but there are continued growth areas. Now, even if we can do this, right, where are we going to get these lungs from? So right now, we work, for example, with lungs from rodents, so mice and rats. We also do a lot of pig lung work. And we also do lungs with, work with human lungs we get from autopsy. So even when we figure out all of the steps and able to recellularize and create a functional gas exchange unit, there still may be limitations on the number of lungs available to do this. And so one of the big thrusts right now is to consider xenogenic approaches, and in particular pig lungs. Now, all of you heard it's been about a month right now about the patient who received a pig heart, heart failure patient, an intact pig heart. And this was from a genetically modified pig that minimized the amount of foreign antigens that would be recognized by the recipient. So we've been working with two companies, United Therapeutics and Revivicor, the same companies that did this pig lung, to approach this with lung diseases, for lung diseases. And the idea here is we're not looking to use an intact pig lung, but rather to decellularize the pig lung and then recellularize it with human lung cells. Very science fiction coming to life type concept, but it's very exciting. And for all the obvious reasons, right? We just don't have enough human lungs to go around. Pigs have a fairly similar, it's not completely, but close enough, anatomy to human lungs. Large reproducible supply, commercial farms. And then again, there are transgenic modified pigs that minimize the number of immune epitopes that would be recognized. So this again is a very exciting growth area. All right, now, what else can you do with these decellularized lungs? And it turns out these are very powerful tools to study disease pathophysiology and come up with new therapeutic approaches short of growing new lungs outside of the body. And one of the things that the decellularized scaffolds retain is the overall complexity of the lung. And so that includes the three-dimensional organization, airways, the blood vessels, the alveoli, the mechanical properties, and again, this extracellular matrix. So the scaffold material of the lung, composition, including structural proteins like different collagens, different bioactive molecules that play an important role in lung cell biology. And what you can see here is this occurs both in a normal lung, but also a diseased lung. So here, for example, is a human lung lobe from a lifelong non-smoker, and the native lung, and then after it's decellularized. And this is a nasty looking lung from a long-term smoker. And you can see how deranged it is, and the like. And once it's decellularized, you can see it's full of black particles and the like. But we can characterize the histology, the cell biology, the matrix proteins and the like, and use this as a tool to study emphysema, COPD, and to come up with new therapeutic approaches. And what this has done is given us a better new set of investigative tools. We can get a better big picture understanding, in particular of the matrix cell, extracellular matrix cell interactions in emphysema and in lung fibrosis. And so we can come up with a variety of biomaterial strategies to model disease-related changes in the lung environment. And these are better models, to my mind, than lung on a chip and other things out there, because you have the three-dimensional structure that you can work with. And then what you can do is really tease out some really interesting things. So the lung is not a uniform organ like the liver, for example. It has different anatomical pieces to it, and those have different mechanical properties, physical properties, and underlying compositional properties. And so for here, in the schematic here in the lower left, for example, you can see down at the bottom, here's a bronchus, so the big airway in the lung, And it's made up of type 1 collagen, type 2 collagen in the cartilage and other substances. And then if you go down as the lung airways divide, you finally get to the alveoli, or the gas exchange portion of the lung. It's a different set of materials. So collagens 3 and 4 and 6, elastin, and things like that. So you can understand this and again, develop understanding of the cell matrix interactions and how those might go bad in disease. We can also utilize techniques such as atomic force microscopy and other engineering techniques to understand the mechanical properties. How stiff are these lungs? Stiffness makes a big difference in the way cells behave. And so you can imagine a normal lung is pretty flexible. It's like a big balloon. It inflates and deflates. An emphysematous lung, COPD, is even more floppy, and it doesn't come back normally, and it's a lot less stiff. In complete contrast, lung of a patient with fibrosis, just by the term fibrosis, is stiff and scarred up. And so stiffness is an important component of trying to understand disease pathophysiology, but also to try and develop new therapeutic approaches. So what else can you do with this? So as part of working with the decellularized lungs, we were trying to develop a new pleural coating. The pleura is the lining of the lungs. And when we were carving up these lungs to study all those cellular and compositional and mechanical properties, we had to develop an artificial pleura, artificial coating. And we went through a variety of materials, including things like agar and agarose and liquid latex and the like. And then we finally came upon an alginate-based material that I'll show you in a second. And then we realized that this investigative research tool might have a more immediate clinical application. And so what we're going to talk a little bit about is collapsed lung, pneumothorax, or bronchopleural fistulas for the medical terminology for these things. And essentially, it's just what it sounds like. So trauma, and this includes military trauma, various lung diseases, and the like. You get trauma to the chest, you get a hole in the lung, one collapses. And what we are able to do is you can put a tube, a chest tube, into the chest. So this goes right through the chest wall, and it's hooked up to suction, and it removes the air from the pleural cavity to let the lung re-expand. But not infrequently, you haven't fixed the wound to the lung. So you've got this hole in the lung that needs to be either patched up or fixed somehow. And this is a real problem. These are old Medicaid database information, but there are a substantial number of patients per year, at least in the United States, a pretty high healthcare cost, and also, of course, morbidity, mortality, hospital stay. So there's an unmet need here. And so here's in schematic form, you see a lung, you damage it, put a hole, the lung collapses, air, fluid leaks out. And if we could come up with some sort of patch, that would be great. So what's out there right now? Surgical repair, you can operate on someone and sew it up, but that's not a particularly pleasant or effective, in some cases, approach. And there's a failure rate, of course, cause pain, complication. There have been a variety of glues that have been tried. So fibrin glue, cyanoacrylates, or super glue, and none of these have worked, basically. There's only one FDA-approved surgical sealant in the United States, something called Progel, which is a mix of polyethylene glycol and albumin. And this is approved for one indication only, so to close up a hole if you have a surgical resection. It's not approved or investigated in trauma. It's not approved or investigated in pregnancy. It doesn't work. You can't use it if you have kidney disease, etc. So basically, there's an unmet need for an effective, safe, easy to use lung sealant. So we again went through a lot of materials, and alginate seems to be the winner here. And so alginate is seaweed-derived polysaccharide compound shown in the upper right here. It's safe, non-toxic. It's fairly widely used in the food and cosmetics industries and growing medical use, wound care implants. It's also easily modified. So the alginate that's out there is relatively inflexible, but it turns out you can chemically modify alginates with simple procedures to make here it's a methacrylated alginate, we call it algma, and it imparts flexibility, which you want if, again, the lung is inflating and deflating and you're putting some sort of coating on it. And then we took a further lesson from nature, which is really, this is cool stuff when you're trying to think broadly. And so mussels, so the shellfish mussels here, you all know they have these tendrils on the tail end, if you will, that allow them to adhere to whatever rock surface they do. And in this schematic blow up of one of these tendrils, you can see this foot. And it turns out that the major adhesive or adherent compound is dopamine. Of all things, it's dopamine. And so it turns out that dopamine is a naturally occurring adherent agent, growing use in tissue and other sealants. And so what we've done is conjugated dopamine to this methacrylated alginate, and we've come up with this easy to use patch. It's easy to manufacture, self-stable, ready to use. And this is a little gory, but this is a rat in which we have opened up the chest. Here's the lung. We make an incision, poke a hole, and then we can seal it up with this patch, right? See this round patch right there. And if we try and quantitate some of the things that we do, here is pressure in the lung, non-injured. You injure it, air is leaking out, the pressure goes down, we put the patch back on, and the pressure goes up. And so we have this patch, and we've been working with the Department of Defense, and we're nearing the end of some longitudinal studies in live rats and live pigs, and we're looking to move ahead with IND discussions to eventually move towards clinical applicability. So this is something, just to reiterate, that came out of studies of materials, matrix, to try and grow a new lung outside of the body. And then it just led to this immediate clinical applicability. And we think that there are other things that can come out of these lung engineering approaches that will have short-term, real-life applicability to lung disease problems. So let's go into another area that's very exciting, which are artificial lungs. Now, many of you have heard about things like LVADs, left ventricular assist devices. And so these are essentially pumps that are hooked up to the patient. And for example, you can wear them in a backpack and the like. And essentially they're mostly meant to be bridges to transplant. So someone who has heart failure needs a transplant, you have an artificial heart that is doing the work for you. So we desperately need things like this for lungs, both as a bridge to transplant for critical illness, including COVID, on which the lungs just are overwhelmed. But also, if you think about patients who need a lung transplant, end-stage lung disease, and they're not going to get a lung transplant because they don't qualify for some reason. They're too old, or they have other diseases that would prevent, etc. So we need devices. So one thing that's out there right now is something called extracorporeal membrane oxygenation, or ECMO. And in the schematic and in the picture on the right, you can see this very complicated ECMO device. It's essentially a gas exchange unit in which blood is removed from the patient, kind of like hemodialysis for kidney disease, and passed through these filters and oxygenators and carbon dioxide scrubbers to take the carbon dioxide out, and then pump back into the body. Now, as you can see, this is a complicated system. It requires specialists to run it. It's not available to every community hospitals. You need specialty tertiary care centers and the like. It's not perfect. The blood can clot. You can get inflammation and infection. But in this lower left-hand chart, you can see that there's been, particularly in adult patients, this dotted line that just starts going off. And this is older data, but it continues that this is more and more widely utilized, if you will. So what can we do about this? So we thought about this a while, and we had this crazy idea based on bird lungs, right? And follow me on this. This is a lot of fun, actually. So here's how a bird breathes. You can see this rooster here. And if you follow, the yellow goes in to these air sacs, all right? And then the air sacs pump, and some of this is through flying motion and the like, pump the air in a unilateral direction through this gas exchange unit. And air goes this way, blood goes this way. It's a perpendicular cross current system. Air doesn't go in and out like it does in our lungs, in mammalian lungs. And so there's no wasted ventilation, if you will. All right. All that air that goes through the bird lung gets in contact with blood. And it's an incredibly efficient gas exchange system. It's not to say that mammalian lungs don't work great. They do. Bird lungs work better. And you can think about this teleologically, if you will, because birds fly, migrate thousands and thousands of miles. And yes, they have wings, but they need an efficient respiratory system. And there's actually a bird called the Himalayan snow goose, which is a bird that can fly over the top of Mount Everest. And so again, you need an incredibly efficient gas exchange unit. So we thought, well, why not take a cue from nature? And if we're trying to make a lung assist device, why not use a decellularized bird lung and recellularize it with human cells? So here we have been working with chicken lungs. And for a larger scale model, we actually are able to obtain emus from some farms here in Vermont. Used to be ostriches, but there are none left. It's all emus. And we can decellularize the small or the large lungs, and you can see it sequentially here. And then we can recellularize them with human cells. And I hope this shows up clearly enough, but this is a photomicrograph of a decellularized chicken lung. And all those little blue dots are human lung cells that are very happy in this chicken lung scaffold. So that's great. So we can create this hybrid system. And then what we do is we put it into a device. And so here's the recellularized lung in this chamber. And then we can hook this up to the patient and then just pass blood through it. And there are all types of pumps and warming devices and things like that included in the schematic. And we've created this avian lung assist device using decellularized bird lungs with, again, recellularized with human cells. Because it's a live system with live cells, it shouldn't clot and shouldn't be inflammatory. And then we could also potentially put the patient's own lung and vast blood cells, if you will, into the system and make this a personalized medicine. So this is really cool technology that is come out of the basic concept of trying to grow new lungs outside of the body for that unmet need. All right, and here's a version in which we would have this device in a backpack, right? And so we envision 3 uses. The first would be for a critically ill patient in the ICU to take place of ECMO. The second would be for ambulatory patient who is waiting for a transplant or will never get a transplant. And then the ultimate is to do this in a small version and actually implant it into the chest, not to replace the lung, but to augment the lung function. All right, so ex vivo lung engineering, powerful new approaches, 3D scaffolds, decellularized scaffolds, organs on a chip. Still a lot of challenges as listed, optimizing materials, manufacturing, et cetera, but these are growth areas. So let's switch gears now and talk about COVID and cell-based therapies for lung disease. So we're going to use acute respiratory failure and chronic obstructive pulmonary disease as paradigms for cell-based therapies. And we're going to talk about mesenchymal stromal cells in particular, rationale, mechanisms of action, the preclinical studies, and where we stand currently with clinical studies. So what are MSCs? So these are cells that were initially isolated from the bone marrow. And you can see here in the schematic, they live as pericytes for the most part. They sit on the outside of blood vessels, in particular in the bone marrow. But you can also get them from fat tissue, adipose, placenta, cord blood. They normally don't circulate. So if you're going to use these therapeutically, you have to get bone marrow or blood, cord blood or placenta, and extract these cells out. And they do have a role as stem cells. They can turn into tissues like bone, fat, or cartilage, but that's not what we're using them for. Rather, they have immune suppressive properties. So they express receptors on the cell surface. They sense the inflammatory environment, and then they release a portfolio of anti-inflammatory mediators. And so this schematic on the right just shows you all the different things that come out of these MSCs. And it turns out if you do laboratory benchtop experiments where you take an MSC and a T cell or a B cell or other immune cells, then you inhibit the functional attributes of those cells as well as their proliferation. It also turns out that they are initially poorly immunogenic. They don't necessarily express on their cell surfaces molecules that would be recognized as foreign. So for example, I could take my MSCs and give them to someone else and they wouldn't necessarily be recognized initially. So you can appreciate the beauty of being able to do this from an industrial standpoint with allogeneic non-matched MSCs. And then there have been clinical trials in a wide range of autoimmune, inflammatory, and non-inflammatory diseases alike. And these cells are approved in Canada and New Zealand for refractory pediatric graft-versus-host disease, a bone marrow transplant complication. And they were approved, they were in the European Union for Crohn's disease. Nothing yet approved in the United States, but that's hopefully in the not too distant future. So let's go back to lung diseases and critical illnesses and see how these MSCs might be potentially applicable. And again, we want to divide into acute inflammatory, including the acute respiratory distress syndrome, COVID, chronic inflammatory, and then the chronic fibrotic destructive disease. And it turns out that there are a huge number of publications in preclinical, so this is mouse, rat, sheep, pig, explanted human lungs, showing that you can induce all different types of lung injuries, acute lung injury, COPD, bronchopulmonary dysplasia, et cetera, and give the MSCs and things get better. And there's a lot of work, and again, this is meant to be illustrative about all these different mediators, angiopoietin-1, VEGF, et cetera, that have been implicated as mediating the protective effects of the MSCs, and all types of mechanisms, cell contact independent, vesicles, cell contact dependent, et cetera, that we're working on to try and understand this. So this has led to a growing number of clinical studies in lung disease. And just to give you the broad base, most of these are still phase 1 safety dose escalation. There are a limited number of phase 2 efficacy studies, but it's important to understand there's no consensus on approach right now. The MSCs, again, come from a variety of different sources, and we don't know which one is best. Autologous, so from the own patient, versus allogeneic, somebody else. Again, we're still investigating which is best. You can gene manipulate these cells, for example, to secrete anti-tumor substances and the like, but that's the minority. Most of these are unmanipulated, and we're still trying to understand the optimal dose and dosing, single dose, multiple doses, and the route of administration. So systemic, you can give intravenously, which is the majority of approaches, but you have the attribute or the possibility of also giving these cells directly down the trachea, so an endobronchial direct lung route. So this big table, a couple years old, but you can see that there are a growing number of trials internationally. The vast majorities are for COPD. There are a lot with this acute lung injury, the ARDS. The majority are in the United States, but certainly a lot in China. And this was pre-COVID. So how does this work? So you collect the cells, and again, allogeneic versus autologous, multiple sources to figure out which one I want to use. You expand these in culture so you have lots and lots of cells. And then again, the major paradigm is you administer these through the blood vessels, so intravenously. So what happens to these cells? So in the first pass through the body, the lung is the first capillary bed that these cells see. And so the cells will lodge in the lung, and they'll stay there for a couple of days, 1 to 2 days, 3 to 4 days, maybe an injury. And while they're there, they will, because of all the receptors they express on their surface, react to the particular inflammatory environment, and then have a broad portfolio of secreted anti-inflammatory molecules, and then they're gone, they're cleared out. They don't stick around. We don't want them to stick around. They don't turn into lung cells. They're not acting like stem cells. they're acting as anti-inflammatory cells when thinking about it. So you can see that illustrated here in this overall schematic as well too. So, so we did the first trial of a stem cell-based therapy for emphysema, COPD, a number of years ago. And this kind of got the ball rolling. So a phase 2 multicenter randomized double-blind placebo-controlled trial, 65 patients, 6 sites, 4 infusions over the course of 4 months. And you can see in this timeline, the infusions, and then we took patients out for 2 years. And the primary endpoint was safety. The secondary efficacy endpoints were lung functions, quality of life. And we also were able to convince the company, because this was an industry-sponsored OSIRIS. And my disclosure is I was an investigator on this trial, no other ties to OSIRIS. But we were able to convince them to measure a variety of anti-inflammatory mediators in addition to routine toxicologies. So what happened? So there were no infusional toxicities. So nothing went bad when we administered these cells to these patients with bad lungs. No serious adverse events in 2-year follow-up. By all these measures, the study was underpowered for efficacy. So initially we didn't see any change in spirometry lung volumes. So lung functions, diffusing capacity, 6-minute walk, standard endpoint measures for these types of tests. But what we did see was a decrease in circulating C-reactive protein, an inflammatory marker that's not infrequently elevated in patients with emphysema. So then we thought about this more and we said, well, maybe there's something there. So again, CRP, a widely recognized marker of inflammation associated with higher mortality, exacerbation outcomes, readmission to the hospital, et cetera. So we said, why don't we do a post-hoc analysis and stratify based on the amount of circulating CRP? And when we did that, we found that in the FEV1 and FVC, again, lung function measures, as well as a 6-minute walk distance, a functional measure, that we actually saw both statistically significant, but more importantly, clinically meaningful improvements in those patients with a high CRP. So that's very exciting, and we're looking to continue. But now, what about COVID? So again, this is a schematic of a normal alveolus on top, and then all the inflammatory things that can happen, both in non-COVID as well as in COVID ARDS. And COVID is different because it's again the SARS-CoV-2 virus inducing this, and there's overlap, it's not completely. Now there's a huge amount of animal data supporting use of MSCs in acute lung injury ARDS, and there are a large number and growing number of trials. There are probably 75 trials listed in the various trial databases right now. All right. Most of these are phase 1 safety dose escalation, some case series and case reports, and all of these have showed safety. All right, so that's a big deal. All right, now the phase 2 randomized placebo-controlled trials are now coming out, and they're looking pretty good. So they are academic ones, University of California, San Francisco, University of Miami, others industry, some listed here. Are in the game as well. Now, again, keep in mind, there is no consensus on approach. What source, dosing, etc., and the like. But what these initial investigations have shown so far, small case series, unblinded case control, mostly umbilical cord MSCs, and that's the convenience issue more so than anything. There are different patient populations. Some of these have looked at the really sick patients on ventilators and intensive care units. Some of these have looked at the less sick patients and the like. So that, again, we're still trying to come to optimal types of approaches and like. But again, outcomes, no infusional toxicities, including pulmonary emboli, no serious attributable serious adverse events, SAEs, suggestion of clinical efficacy. Excuse me. Compared to untreated populations, and the suggestion of anti-inflammatory actions. Again, these are the unblinded trials. Now, the blinded trials. So this is one that was published last year from the University of Miami. And this was a relatively small trial, about 12 patients and the like to each arm, but either got the MSCs, umbilical cord, or the vehicle as control. They got 2 intravenous administrations on day 0 and day 3 after enrollment into the trial. And then the primary outcome were any adverse events, including cardiac arrest or death. And then the secondary outcomes were time to recovery and then a 1-month survival. And you can see the consort diagram here, not to belabor this. They also looked at investigational outcomes with inflammatory mediators. So this is looking good. Here is survival and a Kaplan-Meier survival curve, the treatment group in green, the control group in orange, and it's pretty clear. Serious adverse event free survival, treatment group in green, the control in orange. Now here it's flipped a little bit, time to recovery. So the treatment group had a shorter time to recovery as defined by the parameters of the trial compared to the control group. So this is hugely hopeful, and some of the other trials are coming out with similar data, even though the endpoints may be a little different, the patient population may be a little bit different. And then you're not meant to understand this, but just to see a pretty picture with a lot of colors, with the idea that there was a significant decrease in a number of circulating inflammatory mediators, including IL-6, which is implicated in, of course, in the COVID pathophysiology. Unfortunately, they didn't look at CRP or the D-dimer, but other trials are. They had no difference on viral load. So we're not necessarily killing the virus, but we're protecting the lungs. And that's really what we're hoping to do. So summary and directions in the last couple of minutes. First of all, MSC-based cell therapies and respiratory diseases. Growing evidence supports efficacy in ARDS, particularly COVID ARDS, other lung disease. There are multiple investigational opportunities and multiple areas for collaboration and further technological developments. So then overall, lung regenerative medicine, again, we want to fix these broken lungs, right? And so again, cell-based therapies is really a growth area and very promising. And again, the lung bioengineering, and there are things we didn't talk about, organoids. We talked a little bit about hydrogels as materials and the like, and there are lots of other avenues to try and pursue. So with that, I'm going to put in a plug. We run a conference every other summer at the University of Vermont, stem cells, cell therapies, and bioengineering and lung disease, lung biology and diseases, going since 2005, wide range of government, nonprofit, industry, sponsorship, and participation. And this is, in this world, this is the leading conference to be at. And with that, I want to thank lots and lots and lots of people in my lab, collaborators. We've been very fortunate with funding, both governmental, nonprofit, industry, and the like. We hope to continue this. And with this, to stop here, and delighted to discuss, answer questions, and whatever you'd like to do. So thank you.
Thank you so much, Dr. Weiss. It's a really informative talk. You really cover all the perspective of the regenerative lung disease. So I see there one question coming from the audience, and I do also have a couple of questions. So the question from our audience is, what are some main safety concerns for stem cell therapies, and what cell therapies are currently available?
All right, very good. So one of the major concerns with cell-based therapy is that the cells are going to do something bad, like turn into a tumor or down the road. And so with respect to the mesenchymal stromal cells, the MSCs, this has been looked at very carefully, and there was early data suggesting that these cells may turn tumorigenic, but in meta-analyses of the clinical studies to date, this hasn't panned out. And again, we're still only 4, 5, 6 years into these trials, so no one's going to can tell you what may happen 10, 20 years down the road. But then you take a step back. And for me, in terms of patient populations, let's say those with emphysema, older patients who can't breathe, and you have the conversation that I can't tell you that this may not go bad on you 20 years down the road. And the uniform response is, I do not care, make me better now. All right. Now, you appreciate also in COVID ARDS, when you have someone who's going to die, and you have a potential life-saving therapy, you have to have that conversation and the like. And in the pediatric or neonatal medicine world, of course, it's a huge conversation to have. So that's one worry. The second worry is that when you inject these cells intravenously, then could they clump up in those lung capillaries and form clots, emboli? And this has been looked at very carefully and extensively, and uniformly, there's been no suggestion of clotting, certainly any clinically significant clots. And this is looked at really hard, in particular in the COVID patients, because they have a pro-clotting type of pathophysiology. And we haven't seen that as well. So those are the two major concerns with mesenchymal stromal cells. Now, we didn't talk about embryonic stem cells or reduced pluripotent stem cells. Those both have the possibility of turning into tumors, teratomas, if you will. And we haven't quite overcome that yet. So again, different subject related, but again, these are the types of things that we think about all the time. So I hope that answers the question.
Terrific. So I got one question. So for the organ decellularization process, so certainly it has a really huge potential to engineer, for instance, the whole lungs. But how do you really control, let's say, the cellular distribution process? Let's say during the seeding process, how do you really, let's say, control to, for instance, ensure you have let's say enough cells to recolonize in the shell of the organ. So how do you really control the process?
Well, these are the challenges, right? So working backwards. So one of the strategies to have simply enough cells is to use stem cells. So there are resident progenitor cells, if you will, stem cells that live in the lung. And we can, with increasing sophistication, isolate these and grow these in culture, and then expand them. And so to get the billions and billions, literally, of cells that you would need to put into a lung. And so the second part of the second question is, well, if I can understand, for example, just one example, what the matrix composition in the alveolus, the distal part of the lung versus the big airways and the proximal part of the lung, they have different extracellular matrix proteins and other structural elements such as glycosaminoglycans. And I can understand that how to convince a stem cell that ends up in the alveolus that's going to bind to extracellular matrix protein A, B, and C, that it's going to turn into alveolar cell. And in contradistinction, if I put that cell into the airway, and it interacts with a different set of proteins, that'll turn into an airway cell. So this is where the sophistication of cell biology, molecular biology intersects with engineering, and it's really fascinating. And so there, again, this is where, again, lots of room for innovation and different approaches. We use differential perfusion strategies, differential ventilation strategies. So, for example, if I ventilate a lung like this, then maybe the stem cell will turn into an alveolar cell. If I ventilate the lung like this, maybe it'll turn into an airway cell. So it's that type of combinatorial thinking.
My second question is, how do you solve the vascularization process during the 3D bell printing process? Let's say when you how do you really introduce this functional vas?
That's a holy grail question right now, because again, as you're 3D bioprinting, you have a complex computer guided system to print the blood vessels and the airways and the alveoli, and you have to figure out how to include during the printing process live cells. Okay. On the other hand, you can use a 3D printing process to make the scaffold and then come back and infuse the vascular cells into the blood channels, vascular channels, airway cells into the airways and like too. And in that sense, it's the same general approach as being used for the recellularization of the decellularized lungs, but you've made the scaffold from a bioprinter.
Thanks. We got actually one more question from our audience. What is the proportions of MSCs differentiate inside the human body based on your studies?
Well, for the MSCs to differentiate into, again, their traditional pathways, or if you think about embryonic development, these are mesodermal cells, so they can differentiate into bone, fat, and cartilage. Those are the main avenues, if you will, but they need to sit in a conducive environment for quite some time. And so, for example, if you want to do that in a petri dish on the lab bench, you put the MSCs in a cartilage promoting growth media for a couple of weeks, right? So it takes that amount of time. For us, when we're injecting, and not just for us in lung diseases, but for other people using the cells for the broader portfolio of diseases, you inject them into the bloodstream, and they're gone in a couple of days because they get cleared out of the body after they've done their anti-inflammatory actions, if you will. So can this ever happen? It probably, if there might, you can never say never. If there's some of the MSCs that don't get cleared out of the body and they end up in an area, let's say a joint space, in which the ambient environment is conducive to cartilage formation, then maybe you can turn them into cartilage. And in a separate field, in orthopedics in particular, one of the goals is to be able to inject these cells directly into joint spaces, with the goal not of being anti-inflammatory, although that's helpful, but of actually having them differentiate into cartilage to replenish, let's say, eroded cartilage and osteoarthritis or things of that nature. So again, multiple uses, but again, you have to define your universe very carefully, and specifically in terms of what you're trying to do with the cells.
Very nice. And I also remember you mentioned you're also taking venture money right now for the spin-off ideas, right? Okay. So
we have a number of IP filed, and we have a number of ideas that I think are going to pan out short and medium term for direct clinical applicability, while again we're chasing those dreams of growing that functional lung outside of the body. So we're quite open to discussions about venture and other types of funding opportunities and collaborative opportunities.
Right. I would like to continue our conversation. Let's I'm gonna shoot you an email. Let's schedule another meeting after that.
And again, I'm happy for anybody in the audience. Clearly, I'm happy to engage in conversations.
Wonderful, terrific. Great, thank you so much, Dr. Weiss. Now we come to the end of this great MedLabs talk. So for our audience, if you are interested to reach out to Dr. Weiss, you can find Dr. Weiss information online, right, from the paper, all the contact information.
You can, but I should have included this. So quite simply, my email, DWEISS, at UVM, University of Vermont, uvm.edu.
Can you repeat one more time?
Weiss, DWEISS, at University, V, Vermont, M, so uvm.edu. So
perfect.
Is
that the same email I used to contact you?
Yes.
It's the same email. Perfect.
Yeah.
Wonderful. Great. Thank you so much. It's really insightful informatic talk, and I bet our audience also learn a lot today. Let's definitely stay in touch and continue our conversation.
All right. Well, thank you again for the opportunity, and I look forward to speaking with whoever.
Terrific. Thank you so much.
Thank you, then.